Welcome to our RRP hosted research webinar series. I'm Katrine Erickson, the Executive Director of the RRP Research Program. For those of you who may be new to RRP, we're a nonprofit organization. We're dedicated to supporting patients with RUNX1 familial platelet disorder with a very high predisposition to hematologic malignancies. And we have one goal, and that is to prevent cancer in our patients. We do this through funding research and building and supporting our patient community, and of course, facilitating collaboration like this across our very complex, large healthcare ecosystem. And really, again, with that central goal of discovering treatments or interventions that would prevent blood cancer. I must, of course, also acknowledge the fact that we have wonderful grant co-funding partners in Alex's Lemonade Stand Foundation, Leukemia Lymphoma Society, the Mark Foundation for Cancer Research, and Edward P. Evans Foundation. Without them, we would not have been able to commit over $9 million to date towards research. We started these research webinars in 2020 during COVID, and hopefully you've been joining us all along, and we found them to be really a great way to keep the RUNX1 research community connected and advancing forward. So we'll continue to host these well into the future. And if you yourself are conducting RUNX1 FPD relevant research and are interested in presenting, please reach out. Don't hesitate. I'd love to hear from you, and we can set up a presentation and a research webinar for you as well. Also, if you're interested in applying for one of our grant programs, please do keep checking back with us. We have an open grant program actually right now. You can find it on our website. And of course, if you follow us on social media, you'll see some of these announcements as well. But the one that we have open today is called the Early Career Investigator Program, which is a program co-funded with Alex's Lemonade Stand Foundation. And it offers $180,000 over 3 years. And it's really focused on young investigators who are within their first 5 years of their first faculty position. We'll also be announcing next week, hopefully, a teen science grant program in partnership with several NIH institutes. And so you'll have access to further details, hopefully by early next week. And we'd love to see as many people as possible join that program as well. So that will be a team science where there's an extramural investigator and an intramural investigator working together, again, for the purposes of identifying cancer prevention interventions. Before I introduce our speaker today, I just want to remind everyone that we'll host a Q&A session at the end, about 15 to 20 minutes. And so for that Q&A session, it would really help out if you could use the Q&A icon to submit your questions, and refrain from asking questions in the chat because I won't monitor those as frequently. But of course, if you want to make a comment and share your thoughts on chat, please do so. Okay, without further ado, I'd like to introduce our speaker, Dr. Stephen Nimer, who is a professor of medicine in hematology and the director of the Sylvester Comprehensive Cancer Center at the University of Miami. Dr. Nimer is also an esteemed researcher within the RUNX1 field, and we were just discussing some of his work that dates back to the 80s and 90s. And he's fortunately also a funded investigator through our program with Leukemia Lymphoma Society. So today he'll discuss the crosstalk between transcription factors and tyrosine kinases. Thank you, Dr. Nimer. We're really delighted to have you here with us today, and I'm looking forward to your presentation.
Thank you, Katrin. And let me, for those of you who are looking at the title, I added an extra word, the word methyltransferase. I thought of adding acetyltransferases as well, but I think that the topic of my presentation is how signaling converges on DNA, on chromatin, through a confluence of transcription factors, enzymes, including what are called histone modifying enzymes, but also tyrosine kinases that also impinge on DNA structure, and put all this through the lens of the study of RUNX1 or AML1. And I'm going to start with some historical slides. They have some nomenclature that we don't use all that much anymore, but I thought it could be still instructive because of the history of some of the naming of these genes. So early on, Nancy Speck and David Baltimore identified core binding factor consisting of CBF alpha, which then became known as AML1 and now RUNX1, and CBF beta, and that these bind DNA together. And to activate genes, they bring in acetyltransferases like P300 and CBP. And what this diagram depicts is that these 3 or 4 proteins are actually mutated, translocated, and deleted in various forms of AML and even in ALL as well. P300 and CBP are tumor suppressor genes in lymphoma, and so frequently deleted and loss of function there. And work from many investigators has shown that RUNX1 and CBF beta are both required for definitive hematopoiesis, that the RUNX1 gene is mutated in FPD AML or RUNX1 FPD, and that we and others have shown that multiple enzymes known as histone modifying enzymes, in fact, modify RUNX1, and that includes the protein arginine methyltransferases, the histone acetyltransferases, and the histone methyltransferases. Now, one thing that has driven our lab's studies for decades has been the notion that a transcription factor like RUNX1 can function either as an activator or a repressor. And so you could see here on the left that RUNX1 activates GM-CSF and IL-3 promoters, cooperatively activates SMAD target genes and the TGF-beta activin signaling pathway, activates PU.1 and CD41, represses MIPL, represses P21. And for AML1-EDO, which is not the topic of today's conversation, we have shown and think we know somewhat the biochemistry behind how it can serve as an activator and a repressor. And fundamentally, as would be logical for a single protein, it all depends on what other proteins it interacts with. And so interacting with the RNA pol2, but also bringing in these enzymes, PRMT1, 4, 5, MLL1. And I'm not going to talk about MLL1 except to say that we have shown that MLL1 stabilizes RUNX1 on DNA, and that the mutant MLL fusion proteins destabilize AML1, and therefore that helps promote leukemogenesis. And so the question really still remains, in which circumstances is the activation activity of RUNX1 important, and under what circumstances is its ability to repress genes important? And so what we know from knockout models in mice is that if you delete RUNX1 in adult hematopoiesis, you really don't affect the stem cell compartment very much, but you do cause a rapid loss of platelet production, B and T cells, and not much change in neutrophils or in hemoglobin. So in particular, lymphopoiesis and megakaryopoiesis in the adult are dependent on RUNX1. And this gives us assays, which we hope to use to exploit as we modify RUNX1 function and create RUNX1 proteins that have more activating activity than normal or more repressive activity than normal. And this is relevant in part for the whole RUNX1 FPD syndromes. This paper and others that have characterized the mutations seem to be able to identify certain mutations that are critical for the leukemia predisposition, but not for the thrombocytopenia. This says that the actual level of activity is critical. Half loss of RUNX1 leads to defects in hematopoiesis, and an almost complete loss of RUNX1 leads to increased genomic instability and the development of leukemia. Now, we have shown, this is work done in collaboration with Dan Tenen, Nancy Speck, and many others, that Pu.1 is a major downstream target of RUNX1. If you eliminate the AML1 or the RUNX1 binding region in the enhancer of Pu.1, then you actually can't upregulate Pu.1 as required for normal differentiation. And Dan Tenen and his team, including Uli Steidl and others, have very carefully shown that there's a specific level of Pu.1, and we would argue probably RUNX1, that is leukemogenic. 100% Pu.1 level, normal differentiation. 50% Pu.1, normal differentiation. 0% Pu.1, no leukemia. But somewhere between 0 and 20%, which is done by either deleting an upstream enhancer region from Pu.1 or making some mutations in Pu.1, or the ability of these proteins to downregulate Pu.1 activity, can then create a situation where there's just enough Pu.1 to cause leukemia. And in the sense of the FPD AML or RUNX1 FPD, that's our thought, is that there's a specific level of RUNX1 which is leukemogenic, and if you can up or down regulate that level, you can avoid leukemia development. And so this is shown here. Two things. We have two goals. One, how can you prevent a RUNX1 mutant stem cell from becoming transformed into a heme malignancy? And number two, if you've developed a RUNX1 mutant AML, how do you identify treatments that would specifically eliminate those clones? And so, again, we've talked about enzymes that modify RUNX1 function, can alter either its activating or repressive functions. And how would that work? Is it working on the remaining wild type allele or on the mutant allele? And there are a variety of other strategies. We've been focused on protein arginine methyltransferases or other histone modifying enzymes, because they not only regulate RUNX1 activity, but they can also regulate the activity of P53 and other important genes. So our work and work of others has shown that RUNX1 is extensively post-translationally modified. Some work from Alan Cantor has shown that you can get tyrosine phosphorylation of RUNX1 by the SARC proteins. Some work out of Japan shows that ABL can also trigger megakaryocyte, inhibit megakaryocyte differentiation by phosphorylating RUNX1. The CDKs have a couple of serine phosphorylation sites in RUNX1, and then you get acetylation and methylation and other elements of phosphorylation, all driven by these various enzymes. And to the extent that any of these enzymes are required for a certain level of RUNX1 function, then not manipulating them can potentially impact on RUNX1 mutant AML or the development of AML in a patient with a germline RUNX1 mutation. So our journey started quite some time ago. This is Jinyang Zhao, who's now at UAB. And we took the most common type 1 arginine methyltransferase, PRMT1, did some in vitro experiments, identified a region within the RUNT domain that is methylated, and also a region C-terminal, this RTAM-R motif, where both arginines are methylated by PRMT1. And then we went on to show that using specific antibodies, that if you took human CD34 cells, you could detect RUNX1, no PRMT1, and therefore no methyl RUNX1. But if you differentiated them towards myeloid and not erythroid, the myeloid cells started expressing PRMT1. You saw methyl RUNX1. When you saw methyl RUNX1, then Pu.1 was induced. And so this axis contributes to differentiation. We identified that it was actually the binding of SYN3 that's regulated by this methylation event. So that if you overexpress the wild type PRMT1, you lose the ability to bind SYN3. And if you knock down PRMT1, RUNX1 more potently binds SYN3 and functions as a repressor. So PRMT1 can serve with RUNX1 to activate gene expression. And I won't get into the details here, but one could identify using specific antibodies and CHIP the disappearance or reappearance of methylRUNX1 on its downstream genes. Now, this is relevant, this R206 and R210, because it's equivalent to R233 and R237 using a different nomenclature. And Toshiya Suda, a year ago, published that this mutation is found in FPD-RUNX1 families, and it affects stem cell quiescence. Earlier, we had worked with Tsukasa Okuda in Japan. We generated fundamentally knock-in mice with this R-TAM-R to K-TAM-K, and what we found was a significant impairment of T cell homeostasis. And so I mentioned earlier that RUNX1 controls T cell generation in the adult, and we found the importance of these methylation sites. And so it suggests that PRMT1 and RUNX1 work together to promote RUNX1 function in development. And when this is lost, one has difficulty in differentiation, and one sees some self-renewal and increased quiescence. So I'm not going to get into that earlier work much more, except to say that we've found a whole, what we would call a transcription factor code, like a histone code, where different arginines are modified by different enzymes, leading to different effects. Not only is PRMT regulated in terms of its expression during differentiation, but cofactors are also, and therefore I showed you that the RUNX1 methylation is seen in the myeloid compartment, but not the erythroid compartment. And so this is a fine-tuning of the control of RUNX1 function. We have also shown, and others, that arginine methylation can also impact on DNA-protein interactions. And so combination of protein-protein and DNA-protein interactions that regulate RUNX1 function and the commitment of stem cells to differentiate or self-renew. Now, the other major type 1 arginine methyltransferase is known as PRMT4 or CARM1. We showed, using again human CD34 positive cells, that as you differentiate the cells, that PRMT4 disappears, the level of RUNX1 stays the same. At the level of R223 methyl RUNX1 goes away. And so we focused on this specific R223 methylation event, which is distinct from the PRMT1 methylation sites. And in this paper from Li Vu in our group, we showed that PRMT4, which is highly expressed in the stem cell, blocks differentiation. It does so by generating a RUNX1 containing complex. We don't believe that DPF2 binds directly, but I'll show some data that DPF2, which is in the SWI/SNF family, binds to this RUNX1 complex. And in this case, blocks the expression of miR223, which is required for myeloid differentiation. So when PRMT4 is high, miR223 is blocked. As PRMT1 levels go down during differentiation, you then start expressing the miR223. You can no longer detect DPF2 on the chromatin. You can no longer detect methylRUNX1. And so we think that this is a very important feedback loop where as PRMT4 gets degraded by the miR223 microRNA, you get a positive feedback loop promoting myeloid differentiation. So again, PRMT4 or CARM1 blocks myeloid differentiation of human CD34 positive cells. Now, in that paper, we showed R223 is methylated. We've since gone on in the lab to show that there's also methylation on R319 by CARM1 and not PRMT1 and not PRMT5. And I would just point out for those that aren't complete aficionados on the PRMTs that I mentioned PRMT1 and 4 are type 1s. They generate asymmetric dimethylarginine. There's two nitrogens. One nitrogen has two methyls, the other has 0. Symmetric dimethylarginine, which is generally a repressive mark, has one methyl on each of the two nitrogens, so symmetric dimethylation. And CARM1 is a type 1, it causes asymmetric methylation. R17 and R26 in histone H3. And CARM1 has also been linked to RNA splicing, DNA repair, and transcriptional activation. I would mention that in this case, PRMT4 generated a repressive complex. So don't believe it when you read that these enzymes have only one activity. They can function both as an activator and a repressor of transcription. So we've been very interested in CARM1 and published this article where we made the CARM1 null mouse, a conditional knockout mouse. And we saw no effect at all, almost virtually no effect on normal hematopoiesis. So the CARM1 knockout cells live perfectly well and without any distinction. However, we identified both an inability to transform these cells and an inability to maintain leukemia, and I'm going to go through that quickly. So this is a standard retroviral transduction model. You take ammonito 9A or MLLAF9, you put it into fetal liver cells, inject it into a recipient irradiated mouse, and these mice get leukemia. And so you could see here with ammonito 9A that all the mice are dead around 150 days. But if you introduce ammonito into the PRMT4 knockout cell, which again has no phenotype, appears to be wild type otherwise, none of the mice get leukemia. And even quite unremarkably, the PRMT4 heterozygous mouse, only 20% of these mice will get leukemia in this mouse model. So you have an absolute requirement for PRMT4 in initiating an ammonito-driven leukemia. Quite surprisingly, because of the potency of the MLAF9 oncogene, here you can see also that you don't get transformation of the PRMT4 null. In this case, the hets behave pretty much like a wild type, and that this is a very important finding, that you cannot transform these cells. In part, it's because the cells differentiate, but more importantly here, we establish leukemia. So in this case, we take the conditional knockout cells, but we've not knocked out CARM1 yet. We generate a leukemia with a white count above 20,000. And then at time 0, we give poly-IC and we observe what happens to the mice. So the wild type mice, they all die from leukemia. The het mice, almost again 20%, and none of the mice that have lost PRMT4 can sustain the leukemia. So this suggests that targeting CARM1 is an effective therapy for established leukemia. And so we're very interested in understanding how does CARM1 manifest this activity. And at the same time, we are aware that CARM1 methylates RUNX1. There's a body of literature that RUNX1 is required for a lot of these leukemias, the residual amount of RUNX1, and perhaps that this is a mechanism by which CARM1 is absolutely essential for leukemogenesis. In these models, we see not only differentiation of the leukemia, but it undergoes apoptosis and some cell cycle arrests. So I mentioned that when we overexpress CARM1, we can methylate RUNX1. Here's the opposite. We use some knockdown, a DOX-inducible knockdown of CARM1. You can see here in these lanes, you lose CARM1. when you lose CARM1, here's our RUNX1 R319 methyl. The total amount of RUNX1 doesn't change, but you can see that it is CARM1 that is asymmetrically dimethylating the RUNX1 protein. And here's some of the other known CARM1 substrates. So this 319 is a clear novel site of methylation by CARM1. Epizyme 25654 is a CARM1 inhibitor. And like many of these epigenetic drugs, it takes some time, but you can see here that when you add the Epizyme compound to a couple of leukemia lines, you can see that by day 3, the amount of RUNX 223 and 319 is going away in both of these cell lines. We also looked at similarly at these two cell lines, which are both JAK2 mutant cells, adding the Epizyme compound. And you could see here that about a day later, and certainly by 3 days later, that there is a loss of the R319 and the R223. So knockdown or chemical inhibition eliminates these marks. Now, I mentioned that R223 nucleates a repressor complex of RUNX1, and we've been interested to know what does the RUNX 319 do. So what we did here is we've used bio-ID vectors and have done also some RNA-seq and some ChIP-seq, looking at the binding of RUNX1 and the whole biochemistry. We picked the OCI AML3 line because it doesn't really express measurable RUNX1. And we generated either a wild type or an R319 tag. In these systems, you can induce with biotin, and you can see that there's a lot of the protein there. And the bio-ID system allows you to see what interacts. And we compared bio-ID wild type RUNX1 versus an R319K mutant form of RUNX1 in the OCI AML3. And we found a variety of complexes, including a BAF-SWI-SNF complex. And so we do believe that the methylation of RUNX1 on 319 promotes its activating activity and allows SWI-SNF to bind. Now, cell lines are great. We continue to have a collaboration with Irene Papapetru. We've used her expertise in generating human iPSCs, and in collaboration with her and Michael Karras, we helped with some ChIP for RUNX1, and we've shown now that you can chip the wild type RUNX1 and you can chip the mutant RUNX1 on a variety of genes in these iPSC-derived HSPCs. And so this work is ongoing. I mentioned earlier that there's a lot of work saying that RUNX1 is required to promote the survival of AML cells. And so we are now using the R319 mutants to see the same thing, whether they are capable of sustaining leukemias. Here's another work from the Dutch group showing that the level of RUNX1 is absolutely necessary for amyloido-driven leukemia. And without RUNX1 or ERG in this case, the cells undergo apoptosis. So this does imply that targeting RUNX1 can be an effective way of treating leukemia. Now, for the remainder of my talk, I'm going to focus a lot on CARM1. Again, CARM1 is not required for normal hematopoiesis in the mouse. However, we know from the CD34 work that it's high in the stem cell, it goes down with differentiation, and if you overexpress CARM1, you get a block in differentiation. And so we've then looked at the CARM1 inhibitors on a whole variety of cell lines. And what we found is that cell lines with mutant JAK2 are often more responsive to CARM1 inhibitors than other AML cell lines. And so this intrigued us, this whole JAK2 connection, for a number of reasons. One, Others have identified sites in CARM1 where methylation can affect interaction with transcription factors or dimerization. And there's been a body of work, including some from our lab and Tony Green's lab, that the JAK2 V61F, 617F mutation, which is seen in myeloproliferative diseases and in AMLs, has a variety of effects on chromatin. We were the first to show that JAK2 V617F is in the nucleus, not just in the cytoplasm of cells. And the Tony Green's lab showed that it phosphorylates histone H3, Y41, preventing HP1 alpha from binding to the histone. We showed that it phosphorylates PRMT5, causing a separation from its essential cofactor, MEP50. And it's also been shown that JAK2 V617F phosphorylates TET2, increases its ability to generate 5-HMC. So just to remind everybody that the JAK2 V617F mutation creates a constitutively activated kinase that still associates with other cytokine receptors and can also interact with other JAK members. And that this is seen in P. vera, ET, myelofibrosis, MDS, MPS, MPN, and also in a small fraction of individuals with CHIP. So we did a series of studies and generated some antibodies to Y149 and Y334 sites of tyrosine phosphorylation in CARM1. So you could see here, using these phosphospecific antibodies, that although CARM1 is expressed in all of these cells, that in health cells and in UKE1 cells, there's a very high level of phosphorylation, and there's a more modest degree of phosphorylation in SET2. And these two cell lines have biallelic JAK2 V617F mutations. We also went and showed the specificity of the antibody. So if you add ruxolitinib, which is a JAK2 inhibitor, you eliminate the phosphorylation of CARM1 on these two sites, and also of STAT5 on tyrosine 694. We then went on to show that CARM1 binds to phospho-JAK2, and it does not bind to JAK1, JAK3, or type 2. So it's a specific interaction between CARM1 and JAK2. The interaction itself is not regulated by the phosphorylation of CARM1, but other aspects are. So I mentioned biallelic mutation is important in the ability to phosphorylate CARM1. I would point out, in the absence of biallelic JAK2, you can also get phosphorylation of this via JAK1 or TYK2. So a transphosphorylation event. We knocked out in health cells JAK1 or TYK2, and we'd be happy to make these cells available to anybody. And you can see that when you do that, you markedly reduce by about 50% the ability of the JAK2 to phosphorylate CARM1 on these two sites. So there is some crosstalk between JAK1 and TYK2 that may be important, especially as Ross Levine and others have shown that JAK1 may be an important target in certain cases of resistance. That I'm not going to go through the biochemistry in great detail, except to say that when CARM1 is phosphorylated on these sites, you see that here the wild type CARM1 methylates histone H3 very well. The single mutants or the double mutant do not. If you look at dimerization using a HA tag and a MIC tag CARM1, you see very poor binding based on the Y149 not being able to be phosphorylated. So phosphorylation at Y149 promotes dimerization. And even the nuclear localization is much more potent, much higher with phospho-CARM1. where these things localize primarily to the nucleus. So I showed earlier, and I'm just going to share here, again, in a variety of cell lines, you can detect the methylation of RUNX1 in the same cells that have the phosphorylation of CARM1. So our thought is that JAK2V617F phosphorylates CARM1, increases its dimerization, its movement into the nucleus, and its ability to methylate RUNX1 on R319 and on R223. And you can see here that the epizyme compound does the same thing in a time-dependent manner here, reducing the level of arginine methylation of RUNX1. In this example, we look at the effect of ruxolitinib on the arginine methylation of RUNX1. And you can see here, day 1, day 3, day 5, when you add ruxolitinib, you lose phosphorylation of CARM1 on day 1, and on day 5, you lose the methylation of RUNX1 on R223 and R319. We again turn to the HEL cell model, where we've knocked in single or double mutations into the CARM1 gene. So we've generated here a HEL cell where CARM1 cannot be phosphorylated on either tyrosine residue. And you can see here, when you make a single or double mutant, you lose the ability to generate methyl RUNX1. And so this is a very clear ability now for mutant JAK2 to control the methylation of RUNX1 and its binding partners. And so I showed you that, for instance, this methylation promotes SWI/SNF binding. And so the inability of this HEL cell to methylate RUNX1 would impair the ability of RUNX1 to activate genes. And in fact, that's one of the things that we observed. I would point out that we did some washout experiments. Here you can see that the methyl RUNX1 goes away by day 5 when we've added the CARM1 inhibitor. And then after the washout, both of these reappear. And if you add ruxolitinib, when you do the exact same experiment, you in fact see that you impair the methylation. So if you look on day 7, here, these RUNX1 sites are remethylated. Methylation goes away, now it comes back. But if you've added ruxolitinib, it's not back by 7 days. And so again, the ruxolitinib prevents CARM1 phosphorylation, and that prevention impairs the ability to remethylate RUNX1. Again, indicating crosstalk and also opportunities to use a tyrosine kinase inhibitor to regulate the function of RUNX1. These cell lines are also really interesting. So when you make the mutant or the double mutant, you lose stem cellness. Okay, so the double mutants, actually, I think it's on the next slide, they lose CD34 and they gain CD11B and other markers of myeloid differentiation. You also see an increase in annexin 5, and you see an increase in sub G1 in the double mutant, and a G2M arrest. And so you can use these cells, which have diminished RUNX1 function, to understand exactly how this axis is regulated. And here you can see, based on CD34 and CD45, a marked reduction in the percent CD34 positive, and you see an increase in CD71 cells. So a loss of stemness, and this implies also that perhaps the R223 and R319 mutant forms of RUNX1 may not sustain leukemogenesis, and those experiments are underway. We've done more bio-ID using JAK2 mutant cells with CARM1. We find very clear association of CARM1 with RUNX1. And what we've also done is used a variety of inhibitors. So here we see that a PRMT4 or CARM1 inhibitor on this interferon regulated gene, that you can see an increase in the binding of RUNX1. I would just point out that the PRMT5 inhibitor completely obliterates the binding of RUNX1 to this target. And so we think it's a useful strategy of being able to use these chemical inhibitors to modulate up or down the activity of RUNX1. And those experiments, again, of using diet or other means besides small molecules to accomplish this are ongoing in the lab. And here we showed that the methylation of this R223 leads to repression of these microRNA. And you can see here that if you make a Y149F mutation, you no longer repress the mirror. And so the same for the double mutant. And so this event, phosphorylation of CARM1 on 149, is important for the ability of RUNX1 to repress miR223. We've looked at other RUNX1 target genes, and you can see here that these are target genes down. When you mutate these, some of the target genes that are supposed to go down don't go down, some that are supposed to go up don't go up, and we're still working out the complexity of this situation. So here's what we have. So, sorry. We have phosphorylation of JAK2, which phosphorylates CARM1, which methylates RUNX1, which promotes, in some circumstances, repression of target genes. In the 319, perhaps activation of target genes. And this affects cell cycle progression and is anti-apoptotic, the expression of mutant JAK2. We have found that synergy with using a JAK2 inhibitor and a CARM1 inhibitor on cells with mutant JAK2. And we're examining in more detail the mechanism by which the synergy occurs. And in cells that do not have activation of JAK2, one finds unmethylated, unphosphorylated CARM1, unmethylated RUNX1, and in many, but not all cases, activation of certain target genes. So we've used a couple of cell lines, and these are bliss plots showing synergy scores. We see some modest synergy here in these experiments, suggesting a combination therapy may be more effective. So we are still in the process of identifying how RUNX1 319 progresses in terms of the ability of RUNX1 to sustain a leukemia. But we're very excited identifying a clear connection between tyrosine kinases like JAK2 and RUNX1 function, suggesting that kinase inhibitors can alter RUNX1 function and potentially impact FPD-AML. And so I think I've gone through a lot of this in terms of the biochemistry and the biology. Again, non-phosphorylatable mutants in CARM1 decrease the stem cell fraction, promote erythroid differentiation, and it suggests that we may be able to combine epigenetic drugs with tyrosine kinase inhibitors to treat AML and potentially even prevent its development. So this is a pre-pandemic photo of us actually in Florida, scientists wouldn't take a photo like this without masks, but you can certainly find groups of people here without masks. Most of this work was done by a very talented postdoc, Hide Itanaga, came from Nagasaki, Japan to my lab with help from a variety of people in my lab, some former lab members, and we've used a lot of collaborators here at Sylvester in order to accomplish this work. I'd like to acknowledge support from LLS and the RUNX1 research program, also NCI funding, ACS funding, and I'll stop and happy to answer any questions.
Thank you so much, Dr. Nimer. That was fantastic. We have a question from Wenbin Zhao, and his comment is, fascinating story between JAK2 and RUNX1. Most of the experiments were performed in cell lines. Do you have any data indicative of the relevance in normal hematopoiesis and leukemogenesis?
Yeah, I mean, that's a great question. That we have used these antibodies to show that these marks are found both in normal hematopoiesis and in AML cells. We've not done the biochemistry in AML yet. So with fresh AML cells showing that the JAK2 inhibitor does what it does in the cell lines. So we've been using the mouse models and the cell lines so far.
Great.
Very good question.
You made us a comment towards the end about thinking about using diet, for example, as a way to modulate RUNX1 levels instead of small molecules. So you have me intrigued.
Yeah, so, some work from Louisa Semino at Sylvester when she was at NYU and continues here, has done some elegant work with vitamin C. And we and others have done some vitamin C studies showing modulation of TET2 function. There's some work that we've been doing with Mingjing Zhu at UT South San Antonio, showing interactions between TET2 and RUNX1. And so we're exploring whether vitamin C, for instance, can regulate RUNX1 function. Everyone's aware that ATRA is a form of vitamin A. And we've done a lot of work with LSD1 inhibitors and ATRA trying to trigger more differentiation. And so I think that those are a couple of relevant experiments. We've also done some work with vitamin D on SWI/SNF composition. And so there are the ability to modulate vitamin D levels. And then the last thing I could say, well, it's not the last thing, but the next thing I could say is that there is work on a diet that relates to acetylation, like and methylation, SAM, S-adenosylmethionine, low diets, and whether you can alter at all via the Krebs cycle, how you modify certain histones and other proteins. And so I think we're at the very beginning stages of that. But it's certainly, we're not ready to take tyrosine kinase inhibitors every day. But it'd be interesting to see, exactly how modifying JAK1 or JAK2, because there are people with rheumatoid arthritis, graft-versus-host disease, and other things, non-malignant situations where people are on chronic JAK inhibitors.
Right, and we could actually learn a lot from studying the effects on their blood. It'd be really interesting.
Yeah, what happens to CHIP and other things,
Right, it'd be really interesting. Another question from Julia Maxson. Beautiful talk, Stephen. First question looks like is JAK pathway activation, either through direct mutation or upstream activation, can co-occur with RUNX1 fusions or mutations. Can you talk a little bit about how this regulation of RUNX1 by JAK RUNX1/CARM1 modulated RUNX1 biology works in the context of AML1 EDO or mutant RUNX1? Are there differences? Maybe I'll stop there and we'll do the next one.
Okay. So this is a great question, and in part, if you look in AML at where the most common JAK2 V617F mutations are, it's in AML1 EDO-driven AML. So there does seem to be some kind of a biological event that's occurring, that nature's telling us that these two things cooperate. I mentioned earlier that if you lower RUNX1 activity in AML1 EDO leukemias, you can trigger apoptosis. And so one hypothesis is that the JAK2 both sustains CARM1 and may sustain RUNX1 activity, allowing leukemogenesis to proceed. The other thing I would say is that CARM1 is an AML1 EDO target gene. And if you look in some of our data, so AML1 EDO upregulates CARM1, and CARM1 seems to enhance AML1 EDO activity. And so we believe that that's why even loss of a single allele of CARM1 is so profoundly impairs the ability of AML1 EDO to activate leukemia. We think that first, AML1 EDO as an oncogene is only minimally activated unless there's adequate CARM1 activity. And so what's striking is that AML1 EDO cannot block differentiation without CARM1.
Mm-hmm.
Right, and so we're working on that. It's a great question. And then I see the second question up there.
Yeah,
which is another great question.
The second question is, because not everyone else can read it.
Yes.
The second question is, does CARM1 participate in the regulation of CBP alpha expression by RUNX1?
And the answer is, it is. We know the answer for AML1 EDO. So the answer is yes. Actually, I think we know it for RUNX1 also. So knocking down CARM1 blocks the expression of CEBP alpha. So in the human CD34s, I don't know that we've linked it to RUNX1 specifically, but when you knock down CARM1, you see an upregulation of CEBP alpha, and you see a loss of the methyl RUNX1 at that region. So I think the answer to that is it does participate.
I just have a follow-up question with regard to the loss of RUNX1 methylation when CARM1 is inhibited. Do you also then end up seeing the reduction or increase, I guess, reduction in Q1? So you see that because it's the myeloid differentiation process, right?
Yeah. So those experiments so far have only been done in health cells. I'm not exactly sure how much Q.1 is there, but I'm going to text the postdoc while we speak to see if we can probe that Western blot for Q.1 and see. You would predict that it would be down. On the other hand, we see some changes, but in almost all circumstances, the R223 methyl and the R319 methyl are occurring coincidentally. But our data so far suggests that the 319 plays an important role in activation by RUNX1, and the 223 may play a role more in repression. So it's probably sort of a fine-tuning rheostat, and there's different proteins that are bound to those sites, and there's going to be very much context-dependent effects, I believe.
Interesting. We have another question from Connie Bonifer. The inability of CARM1 knockout cells to transform and instead differentiate is intriguing. What is happening with normal hematopoietic stem cells over long periods of time?
Yeah, it's a great question, Constance. So what we find is, first of all, CARM1 is dispensable, but we do the absence of CARM1, we see an expansion of progenitors over time. So we see a modest loss of stem cells, a modest increase in progenitors. And so somehow the normal cells, we've not created a double knockout, let's say CARM1 and PRMT1, which are, again, maybe lethal, but somehow the cells can accommodate over time. And we followed the mice for over a year. And all we really see is a modest increase in progenitors.
Great, thank you. I'm just waiting to give folks one more minute if they have any other questions. I'm curious, could you speculate in the case of germline RUNX1 patients prior to malignancy, if you were to treat with a small molecule like ruxolitinib or other JAK2 inhibitors, what is the underlying mechanism that would prevent leukemogenesis from happening? Because you're essentially just pushing differentiation by using ruxolitinib?
Yeah, I would defer. I think, seeing who's been asking questions, there's a lot smarter people than I on the call, but I think the answer won't be as good as the question. But what I would say is, I would think that anything that, if you could promote a bit more RUNX1 activity, you're better off. Whether that's it's activating activity or it's repressing activity, I don't know. But my sense is that you reach a critical level of RUNX1 activity, it affects the genome, and then you start generating other mutations, whether it's B-core or a second RUNX mutation or something like that. And so I wish I knew whether you're promoting repression or activation by RUNX1, but I think that you need more RUNX1 function. Mm-hmm. And so we're hoping with these single and double mutations in the mouse models to see which one of these would increase leukemogenesis and which one could decrease leukemogenesis. Because they're both sites, but at the moment they seem to have somewhat different roles in RUNX1. And so I guess if I could somehow boost 319, right? Mm-hmm. That would be a good thing, and somehow repress 223.
Yeah. Which means that we have to be, we'll have to be thoughtful of each different germline mutation, because the dosage of RUNX1 activity might be different, and that's important too.
Yeah, the other thing which people, and again, maybe an easier way with these, understanding what are the E3 ligases, is that if you could block the degradation of RUNX1, some of these mutants have loss of function, some are dominant negative, but if you somehow could restore some RUNX1 function via blocking its degradation, perhaps that would be beneficial also.
Another approach from the healthy allele. Yeah. Well, we're 1 minute over, and I want to thank you again so much for the presentation, Dr. Nimer. It was fantastic, and lots to think about, lots of learnings, and thank you everyone who participated. And hopefully we'll see you all back at our next research webinar, hopefully in January.